This video is going to focus on the final step of animal viral replication and also talk a little bit about different types of animal virus infections. So on the last video, we focused primarily on biosynthesis, which was the fourth step of viral replication. And then remember, just like bacteriophage, animal viruses, once they've produced enough viral nucleic acid and viral proteins, they will spontaneously assemble. And that's step 5, called maturation, also called assembly. And so the final stage is release. And the mechanism of release is based on whether or not the virus has an envelope. Okay, so naked viruses are released by lysis. We say that these viruses are cytocidal. They are going to kill the host cell in order to exit. Enveloped viruses are said to be cytopathic, and they are going to leave via budding or exocytosis. So let's take a look at this. So we did see a picture about viral budding at one of the earlier videos where the virus is kind of like pushing its way out of the cell, and in the meantime, it is taking part of the host plasma membrane with it. And so here on the left side of the image, you can see where the virus is spontaneously assembling and starting to push on the host cell membrane. And then eventually it will be able to push its way out, forming an intact virion. In this case, that has the nucleic acid, the capsid, and an envelope. Okay. So some viruses, as you can kind of see here, are tethered to the host cell when they bud off from it. And so neuraminidase is actually an enzyme shown here. It's actually one of the glycoprotein spikes, is able to cleave this bond so that the viruses are released from the host cell and able to go out and infect new hosts. And then here is a picture of exocytosis, which is the process of releasing the intact cell. And they show you the process for different viruses here on this picture. And I'm not going to ask you to remember how Hepadnaviridae, the rotavirus down in like the kind of the bottom middle region, how that's different than the Bunyaviridae, right? Or the Arteraviridae or Coronaviridae even. But do notice how viruses exit the cell very differently. Okay, so the general process, as you can see on the far left, some of them are, the viruses are associating with the endoplasmic reticulum, in which case, and so let me highlight where I'm talking, just because there's a lot happening in this figure. So I'm looking over on this end down here. Okay, so this virus right here is interacting with the endoplasmic reticulum. Okay, and it kind of buds off from it. So notice the intact enveloped virus is inside this little vesicle, okay, which is it's going to kind of follow its way out. And notice that once it gets to the end over here, it almost looks like the cell is kind of like spitting it out, right? Just kind of pushes the virus outside of the cell. Okay, now other viruses you see over here, we have the herpes viridae, pox viridae, the Bunyaviridae, right? They are interacting with the Golgi complex. Okay, so these guys are all acquiring their envelopes from an organelle. But remember, organelles are membrane bound, so they're still acquiring that host membrane. And so in this case, from the Golgi, they're then transported here to the plasma membrane where they are released from the cell. Again, this overall process of releasing something rather large, like a virus, is called exocytosis. Okay. All right. So here I just want to show you how different viruses are in terms of the host cell that they use and also what their burst size. So how many new virions are created and released from the host cell. And notice there's quite a bit of variability within the animal viruses, which are in the top three numbers here, and then the bottom three are for bacteriophage. And there's quite a bit of variability, right? Can you imagine one influenza virus can actually, when it infects a host, can produce up to 10,000 new viruses, right? So that's why sometimes when you get infected by a virus, it feels like you've been hit with a ton of bricks. Okay, so categories of animal virus infections. We have acute infections, which are basically the virus comes on strong, right? It hits you really quickly, very rapid onset, but it is over and cleared relatively quickly. It's got a short duration. And a lot of times, the reason for this is because they do result in the death of the host, okay? And a lot of times there's a larger burst size associated with these infections. The persistent infection is where a lot of time the viruses or their genomes are present within the host for a very long time. And oftentimes they're released by budding. And these are called persistent infections because they last for a really long time, years or even a lifetime. But there's not always symptoms. If you have a virus for life, that doesn't mean you are always feeling the symptoms of that virus. All right, trying to go to the next slide, the last slide for this video. So we also have persistent infections, right? So we just talked about are gradually over a long period of time. And then latent infections, which is where there's a delay between the infection and the actual lytic process. This is where they kind of hang out for a while. We talked about this with HIV, those retroviruses, where they actually, even though it starts off having an RNA genome, it's able to convert the viral RNA to viral DNA, which then is integrated into the host, and it just hangs out for a long time. And so with that, they can be with you for life. And whenever they exit the cell, they actually are reactivated. That's what it's called when you kind of get those symptoms again. And these guys cannot be eliminated. Okay, they remain asymptomatic for very long periods of time. But I want you to also look at this graph here. So we've already kind of talked about it a little bit. You saw the one-step growth curve graph. This is the same thing, but talking more about persistent and latent infections. So again, you have that initial inoculation period where viruses are attaching to host cells. But remember, once those viruses get inside the host, what do? They uncoat. And when they uncoat, they are no longer intact infectious virions. So that's that initial decrease that you see happening there. During this eclipse period is where the virions are, getting into the cell, and then eventually they're going to start going through biosynthesis and maturation. And you're going to see this increase in virions, and then they are going to be released and move on to infect new cells. All right, y'all, that is the end of this video.